Well, it's great to be back at the ISB. At the time when we started with 4 or 5 faculty members, we couldn't dream that the term systems biology would even catch on. It was pretty fresh then, and it's amazing to see what has happened over the years. Now, the emerging technology that I've been working on for 40 years is flow cytometry, always on the verge of a breakthrough. And I'm going to report today to you how I use this technology in studying phytoplankton communities in different areas of the world. Now, what flow cytometry allows me to do is to analyze every plankton particle in the ocean or any watery sample, and then try to identify them, build up the community from those observations that identify particles, that determine some aspects of their metabolism, and then try to relate the phenotype that we see in these assemblages to the genome, where the genome is just a list of potential actions. What we can determine by cytometry is to see what's actually manifested, what's actually expressed of those actions. And then flow cytometry also allows us to dissect the population. We can get them viably in our hands and do experiments with them. Now, we all stand on the shoulders of those that went before us, and we easily step on the toes of those that are next to us. So before I do this, I should give credit where credit is due. Ann Thompson here at the ISB has been working with me. I work with the Seymour team in Hawaii. Ken Doggett and Dave Carl are my main collaborators there. And then at the Instituto Milenio Oceanografía in Chile, I work with Osvaldo Ulloa and Gadiel Alarcón on the coast of Peru and Chile. The system that I'm interested in is one of the biggest biological systems one could study. It's the ocean. 3/4 of our planet are covered with water, and we know very little about it. And yet all life originates from that place. And we're also modifying the ocean at a very rapid speed. We're polluting it. We are tinkering with the pH by dumping CO2 in it, and we know very little about what's happening there. Now, if you look at it, we tend to see the large things first, and we think we know a lot about oceanic life. With microscopy, we can see smaller organisms, but the real important components in the oceans are these very dim spots that you see down here. These are the photosynthetic organisms, microorganisms, 1 micron or smaller, that have different pigments and that are the source of all life, of all carbon, all organic carbon that's on our planet, basically. And these things have been instrumental in shaping the planet and our world as we know it. 2.5 billion years ago, the first cyanobacteria appeared. They could produce oxygen by photosynthesis. The original billion years, production equaled consumption. All the carbohydrates formed were burned again, so oxygen released was used. And it wasn't until organisms appeared in the ocean that decorated themselves with calcite and other heavy molecules, heavy materials that sunk to the bottom of the ocean and got buried, that all the organic carbon got buried in the ocean. Then there was an excess of oxygen, and oxygen was released in the atmosphere. Then it took another billion years before all that oxygen was absorbed by iron, and when all the iron was oxidized, eventually 500 million years ago, the more complex animals that we are could appear, because we have a very expensive energy budget and we need oxygen to run our lives. So these things not only formed our atmosphere, they formed our mountains. The coccolithophores are the basis for the Himalayas and other mountain ranges. They also made it possible for us to exist and do what we're doing. It's a very, and in many ways, they shaped our planet the way we know it. And yet, we know very, very little about them. The original approach was top-down approach. NASA always published these beautiful chlorophyll measurements done by satellites. And when we started looking at the ocean through the satellite eye, we concluded that all the important things were happening in the Arctic regions. After all, chlorophyll is the basis for this whole process. This is the only chlorophyll that you really spot with a satellite. What was missed in that picture is that most of the chlorophyll is actually in the tropical oceans, but you don't see it because it's locked up in these very, very small cyanobacteria, smaller than a micron in diameter. They are very sparse because the oceans are oligotrophic. You cannot support a high density of life. But through the top 200 meters of the ocean, these very minute organisms form a very large reservoir of organic carbon. About 10% of the Earth's organic carbon is locked up in the species. It's estimated that 1 out of 4 oxygen molecules that you breathe are produced by Prochlorococcus. Now, this organism wasn't discovered until the late 1980s, and it was discovered by flow cytometry. And this discovery was so enormous that so monumental that it has fascinated me ever since. Flow cytometry, very quickly, we inject a sample into a stream of water. The stream is narrowed down to a very small orifice so that a very thin thread of liquid emerges from this hole here. The particle suspended in the medium passes one or more laser beams. Every time a particle is in the laser, you see a light flash, fluorescence, or scatter. You can measure the fluorescence and the scatter and count the particles and identify them on the fly. Now, and then if you look at samples from the ocean, you'll see these clusters of fluorescent and scattering particles that then can be identified by sorting them out. It's possible to take that water jet, divide it into drops, and deflect every drop that has a particle that you identified as being interesting, and then you can get those particles separately in hand. They're still viable. You can do experiments with them. You can identify their DNA. You can do all sorts of things. So this is the technique, highly analytical, tens of thousands of particles per second. And then if you see an anonymous group of particles that are somehow of interest to you, we can separate them and do experiments with them. So my goal is to develop flow technology for phytoplankton analysis so that it's possible to actually compare the data between different groups and collect it on different sites of the world. Standardization of these fluorescent techniques is quite difficult. And why is this necessary? Plankton is very, very small, about only 2 to 3 times the wavelength of light. The particles are only about 1% of the volume of a small blood cell. So in scatter and fluorescence go down exponentially with the size, so they're hard to measure, hard to see. And not only that, in the 2.5 billion years that these particles have been around, they have learned to utilize nonlinear mechanisms to harvest light. They're very, very efficient, and maybe by studying the nonlinear effects on these particles, we can learn more about how they physiologically function and are capable, despite their diminutive size and the small amounts of chlorophyll that they carry, to be so efficient in photosynthesis. So that's my goal. Now, to give you a few examples, while nonlinear mechanisms are important, if you take a plankton sample from the Barents Sea and you study it with traditional flow cytometry criteria, you measure scatter, forward scatter versus chlorophyll fluorescence, you might see this rather uninteresting cloud. Maybe there's 2 parts to it, maybe 3, it's hard to say, but you basically see an amorphous cloud of chlorophyll-containing particles, and it's hard to distinguish between them. If you do the same thing and you look at energy transfer at different colors, you suddenly see that this cloud dissociates into very precise needles on different places on diagonal of this plot. So suddenly we go from an amorphous mass to distinctive needles. And if we increase the number of colors that we're using, more lasers, then suddenly more and more needles become apparent. So by employing technology that isn't used in any way in biomedical research, but looking at energy transfer between different fluorochromes that natively are present in these algae, we can start to see a very heterogeneous group of organisms that before that looked quite homogeneous. And in this analysis, we can see about 19 to 20 different clusters, but before there were only 1 or 2. So that's the sort of approach that I'm trying to develop, in which we look at new parameters, new ways of looking at fluorescent particles and figuring out that we can see some reproducible heterogeneity in them. The other thing that is not well understood for these very small particles is light scatter. Light scatter of very small particles goes with the 4th power of the linear dimensions. It's demonstrated here with beads. And it's quite a different parameter from side scatter, where we see an oscillating curve that goes with the square of the linear dimensions, and there's destructive and constructive interference that is visible in this pattern. Unfortunately, if we look at the very small organisms like Prochlorococcus, they seem to obey the same optical laws. So these are beads, plastic beads, and if we take these slopes and put them onto a slide of Prochlorococcus particles, we see that they seem to follow the same relationships, and they also have the curves that correspond to interference with the light waves. So in trying to understand what's going on, maybe we can also tell something about the size and the structure and the things that are typical for these particles. Now, what I've done over the last 10 years or so is to help create mobile seagoing cell sorting labs. If we want to do this right, we need to do it on living organisms that we pluck out of the ocean, and then we try to build into these labs the capabilities of nutrient and analysis and other geochemical parameters, and we try to incorporate depth profiling pumps that go up and down in the ocean, so that we can read a whole profile through the euphotic zone and figure out what the organisms look like. This is what my labs are. This is a van, a shipping container that has a complete lab inside. We can put it on board of a ship. This is the one in Hawaii. It's pretty much like a prison cell. If you go to sea, you're there 12 hours on, 12 hours off. But they're very comfortable. This is the lab in Chile, where we have all the microscopes. We have a good audio system. Music is very important. And have all the scientific equipment needed to do our studies. We can hoist them on board of shipping vessels or on fishing vessels. This is on board of a trawler, in a Norwegian trawler on the shipping on the deck where they let their trawls out. And then we can take them to sea. This is about 500 kilometers from the North Pole, where the shipping containers, my comfortable home for a few weeks away from home. I have access now to 3 of those places. Station Aloha in Hawaii, where we study the deep eutrophic ocean. Under the coast of Chile and Peru, where there is oligotrophic, eutrophic waters and interesting layering of the ocean. And then we have access to a facility like that in the Arctic. So to start with a simple system, the deep nutrient-poor waters north of Hawaii, very deep, 4 to 5 kilometers deep. Not a lot of light visible. With the satellite, you see nothing. But if you go in, you see a pretty bleak picture, pretty meager picture. There's one dominant cluster. This is the surface water. This is forward scatter size, roughly, versus chlorophyll content. 6-decade resolution here. This is Prochlorococcus, the organism I talked about before. 98% of particles belong to this group. 2% of the particles are in other cyanobacterium, Synechococcus. This one has phycoerythrin, that's why we can recognize it. And then all the other stuff that preys on it and feeds on it is a very small minority. So in this is a very simple system. There's basically only 2 dominant species, and one of them is very, very dominant. Now, these organisms are mostly studied on cultures in the lab, and you look at them and they describe this is the way they are, and there's no indication of plasticity, of an ability to change their shape or form in any way from those lab studies. However, if you go down, and we're now going down at 5 meters a second, where we take different samples, you see that they're not static at all. They, as you go deeper, they change gradually. They develop more chlorophyll in the beginning, and then they start moving along this diagonal line. They become bigger, get more chlorophyll. You see a double peak at sometimes, 2 populations. There's also an orange component that develops as you start leaving the euphotic zone and get into the deeper, darker waters. So these, and in fact, if you show me one, if you're experienced with these pictures, and you look at one of them, you can say within 5 meters where that sample was taken from. So if we go to 200 meters of ocean, every 5 meters in that ocean layer, the presentation of these populations changes quite dramatically. So what does that mean? What's going on? And this is another way of looking at it. We can then generate depth profiles. We can plot the chlorophyll content, the orange signal, assumingly phycoerythrin, the scatter content as a function of depth. We can count them because we do this in a volumetric way. We measure 100 microliter samples, count the different species, and we can measure the light intensity. Again, this is a log scale. And then one of the first things that we notice is that in the top layer, where the mixed layer, where the ocean is mixing with relative efficiency, where there's a lot of light, there's at about 100 meters in Hawaii, there's only 1% of the light left, that the chlorophyll content of Prochlorococcus is inversely related to the light intensity. So loss of light is compensated by getting more chlorophyll, which seems to indicate that maybe in this zone the productivity is constant, something that is not at all accepted by the community of oceanographers at large that always take chlorophyll content as a measure of productivity. In fact, what we see here is that productivity is not related to chlorophyll at all. It's the other way around. Chlorophyll is compensating for loss of light. Then in the deeper waters, we see an exponential decay over 100 meters from over 2 or 3 orders of magnitude, a beautiful linear decay, and then finally at 200 meters, the species are gone. In a way, this is also related to the genome. And Thompson has done real-time PCR as we bring in up those samples. And here we see the 4 PCR markers for different classes, different types of Prochlorococcus. As the distribution goes, we could wonder if there's such a heterogeneity, we're either dealing with a huge variation in genomes, or we're looking at a limited number of genomes that adapt to the layer that they find themselves in. Well, the last seems to be the case. We find a different genotypic markers quite broadly distributed over these layers. The so-called highlight Prochlorococcus. Highlight should be taken with a grain of salt here, because they cover full daylight to 1% of surface light, and in all cases 100-fold light intensity are available. Then close to that thermocline, we find that 2 other species start to dominate, but we also find them in the higher well-lit and not so nutrient-rich zones. So the separation of these ecotypes is not absolute. They seem to be dispersed much more homogeneous than one would expect based on the laboratory management that classify them as highlight, low nutrients, and etc. Another place where I go is at the currents off the coast of Chile. Cold nutrient-rich water comes from the poles, and then there's an undercurrent of ocean water where that takes about a year or so to get from the Antarctic to this place, and all oxygen in that time is being consumed by anaerobic processes or aerobic processes, and that water is total oxygen free. So there we have a very interesting habitat, cold nutrient water at top, no oxygen at the bottom, and through that gradient we see a diversity of sea life. This is Synechococcus, which was only 2% of the Hawaii. It dominates here, and with energy transfer we can see the different types of Synechococcus, and at every sampling site that you go, you find a different composition of these Synechococcus populations. So there's here an ecological diversity that we can't quite explain. There's a simple current coming up, and yet at different sites we find different compositions of the Synechococcus populations. It's a very interesting depth profile. The Synechococcus here is in the red dots, constant over the first 50 meters or so, the numbers, and then they decay exponentially, just like Prochlorococcus at Hawaii. Prochlorococcus comes up, is constant, but now in the minority, then comes up where Synechococcus disappears. They seem to be specializing in this region. They all disappear then, and then where the oxygen disappears, the anoxic zone comes, a second population of Prochlorococcus appears. Now these Prochlorococci are all very different from the ones we see in Hawaii. Genetically they're different. They haven't been categorized very well, but we are dealing with very different genomes here. We can also follow this, change in the environment by looking at nitrogen. Ammonia is high in the region where there's high productivity, represents decaying material. Then the nitrates come up where life cannot consume them, and then in the anoxic zone the nitrates are replaced by nitrites, and we see a totally different population. So what was thought to be a homogeneous ocean that you could just study with a satellite from above now turns to about a very heterogeneous and layered structure supporting all sorts of sub-ecosystems that are very, very different in their metabolic processes. Very quickly, I only have 5 minutes left. I'd like to reinforce this view that the ocean is much more complicated than we thought. The cyanobacteria are not just passive bystanders that proliferate or don't proliferate. They're actually actively interacting with the physical factors in that environment. We happened to be an ocean where a big storm appeared. You can see that here from the temperature curve. Before the storm, the black line represents a very stable temperature and gradual temperature curve. At the storm, the top 100 meters is all mixed, all the same temperature. Despite this huge movement of water, this was a major storm, the numbers of Prochlorococcus do not change very much. But what we do see is that the lower thermocline waters move up and down, slosh up and down about 50 meters in about 6 hours. There's huge waves, subtidal waves going on. And this may be, I think, the key factor in explaining some of the organisms. Now, the amazing thing was that 24 hours before the storm, I thought my instrument was on the blink. We got profiles that didn't make any sense at all. I discarded these data originally. I thought something was very wrong. Our normally very nice smooth curves were very jagged. And then we noticed that these strange outliers corresponded to salinity changes in the ocean, and that big layers, masses of water had slid under and carried Prochlorococcus and Synechococcus populations from different elevations, and we got a kind of a layered mixing up that we could follow by the chlorophyll content. So this really reinforced the notion that this system is extremely variable, that big water masses get translated, and that you can follow that by following the fluorescence profiles of these algae. Here's another example. There's an inversion here between layers. You see it as in total chlorophyll fluorescence. You also see it in the salinity and temperature discontinuities, and it also comes out in the numbers that you count. So very quickly, and this is another one where there was a small, again, a small discontinuity that corresponded to a salinity layer. So we can actually follow the physical ocean by looking at the physical properties of these of the prochlorococci. Now, the amazing thing here is that over 250 samples that were taken before the storm and after storm, from the surface to 200 meters, the chlorophyll content of Prochlorococcus correlates to the chlorophyll content of Synechococcus, and absolutely. This is over 2 orders of magnitude. If you know what the chlorophyll content of Prochlorococcus is, you can tell what the chlorophyll content of the Synechococcus is, which tells me that this is not genomics. This has to do because the two species, the two organisms are wide apart on a genome scale, but they must listen to the same stimuli in the ocean. They must see the same physical factors that determines how high their chlorophyll expression level should be. And this, in my life as a biologist, I never had data that correlated so nicely. So to close up, plankton communities look stable, and we've been looking at them as if Prochlorococcus from Hawaii could be compared to Prochlorococcus from Chile or other places. Not true. There's a lot of diversity in phenotype, at least, that is correlated with different layers in the ocean. Prochlorococcus, Synechococcus actively respond to changes in the environment, and they do it in very similar ways. But that change must take place over different generations. What I forgot to tell you is that they divide every 24 hours. So 5% of the world's carbon goes through this compartment every day. 20 days we have replaced all the carbon in the ocean. And yet we can see events and deeper layers that still, or populations that look like other populations that must have slid under that water a few days before. So they have some memory about what they're supposed to do. Chlorophyll is inverse to the light intensity, which seems to suggest, over 100-fold range, seems to suggest that chlorophyll maximum is not important, and seems to suggest that there is a constant productivity in the well-lit region of the ocean. Now, the big difference between the deep ocean and the surface ocean that we see in that storm when the thermocline moved up and down is that the light exposure in the mixed zone will have a 24-hour periodicity. However, you go lower, you get sometimes exposed. When the storm comes, you are brought up 50 meters and you see light, and then you go down again. That must have a completely different kinetic behavior than when you're in the surface zone. And that's what I think is the main difference between the high light and the low light prochlorococci. The low light prochlorococci are missing the clock genes that the high light have, and I think they're adapted to an uncertain, they're opportunistic, they are not prepared for a 24-hour cycle, but they're able to use light as soon as it presents itself, and then do the business. I didn't get to this, but there are some other interesting physical processes that you can follow by following these organisms. And then I'd like to leave you with the impression that the interplay of physics and plankton dynamics is as important in understanding the genomics and the expression of the genomics of these organisms and that we often tend to neglect that the microorganisms live in the real world and must have adaptations that respond to changes and challenges of the environment. And with this, I would like to thank the organizers again for inviting me, and I'll stop here.
How heterogeneous are those two big populations that you talk about?
The two big, well, that depends. If you look at mutations, Penny Chisholm now says she has thousands of different genomes. So it's in the eye of the beholder, I think. If you're just a microscopist or a flow cytometrist, you say there's one population. If you go for every mutation, there's probably an unlimited number. Now, what's relevant there? That's the important question. That's what we're trying to answer here.
Back here. Hello, Ger. Lovely work. Question, and I think maybe you answered it at the beginning, but I'm assuming that Prochlorococcus isn't sedimenting, meaning that it stays
Well,
where you put it, or is it sedimenting?
That's very interesting. Particles of 1 micron cannot swim, never. It's impossible physically. And what we discovered here is that we see them go down very rapidly, and by these mixing of the ocean layers. And what I think is going on, a process that has been recognized but not really being found very important in Hawaii, is salt fingering. We get high salinity, warm water coming from Mexico, coming in, cooling down, and going down quite rapidly. Personally, I think this is what explains the exponential decay curves. Exponential decay curves always point at physics somewhere. And I think that salt fingering is much, much more important than what it has been given credit for so far. And actually now the physical oceanographers are going back in Hawaii and actually starting to look for evidence that this may be a much more important mechanism than they thought. So in this case, biology is actually providing us indicators to look for new oceanographic movements.
So I have a question here, which is around, so it seems to me that because they are so reflective of the environment they're in, right, they actually are going to be sentinels in a way in the change of the environment. Any sense of what it'll take to actually get to a baseline curve to be able to watch these series of populations as climate change impacts? And also might they be obviously sentinels of both climate change and other environmental impacts?
This is, of course, what I hope is happening long term, what we're trying to achieve. The place in Hawaii, we go to almost every month, and we have 25 years of observation there. Yeah, if we can read the biological fingerprint here, if we understand what they are doing and what they're responding to, yes, maybe we can use them as indicator species that tell us the acidity is reaching critical levels, the temperature is getting too high, and what have you. But right now we know so little about the basic physiology of them that we're not there yet. But we could get there if we have enough data and enough observation stations. Because again, 3/4 of the world covered by only 3 labs that go out to sea for maybe 30 days a year is not.
Right. And do you see a future where, in fact, they might be able to be manipulated in a way that could be, in the sense, therapeutic for what might be environmental changes?
I would be very hesitant to do Earth engineering, but yes, I could envision that there are people that would like to do that, yeah.